Okie dokie. So here we go. We're going to move on in our biochemistry unit. So there's absolutely nothing ever now that we don't understand about water. And we're going to move to carbon. And again, so this is 4.1 and 4.2. We're going to find that just like the way water is shaped is the reason why it acts like it does and has the properties it has, we're going to find that because of the way carbon's made and shaped, it also has properties that are unique to it. So, like we did the other big monstrous stitched together bunch of videos, that's what this one's going to be. So we're going to take some notes, watch some more videos, take some notes, a little bit of this, a little bit of that. You know how that goes. Okay, so I don't usually ask you, because they don't usually ask you, to memorize what anybody's experiment is. But what they do is they ask a question, so they're talking to you like you just know. So sometimes it's good if you actually have it in your notes and you've seen it before, so you can place it in your head. When you go back in your head, you're like, oh, I've written that before. I don't know about you, but I can see it in my head. Doesn't always mean I remember it. But so we're starting off with just some experiments. Like this experiment was pivotal around recognizing that carbon-based molecules could exist on this planet from the conditions that were originally here. That's the sum of it. So it's called the Miller, but it wasn't just him. He had a partner, Urey, but they don't usually ever talk about him. I don't know why. It was, so this is an experiment. It was in the 50s. So I know that, you know, we watch Happy Days or talk about things that are going on back in the 50s, watch Back to the Future, or all these things. That didn't seem like that long ago. You know, it's 70 years ago, but, you know, people like my mom grew up in the 50s. So it doesn't seem that long to me. But here's the difference. We knew very little about molecular structure. We didn't know what DNA looked like. We were working through a lot of things. You couldn't sequence DNA. It was just different back then. So the question that was asked was this. Originally, we had in the atmosphere and on the planet, the conditions originally were methane, not the most pleasant, but the simplest carbon, hydrogen, so hydrocarbon molecule. You had ammonia, so that would be a source of nitrogen, and hydrogen, and water being a source actually of oxygen. So you had all of these components, and they put them in, usually it looks like some hodgepodge thing that you'd see in a picture, or I don't know, but they put them in a flask, they zapped it with some electricity to simulate lightning, and these molecules were able to have the energy that it took to create bonds and to form more carbon-based molecules in variety. So all of these simple, this is what conditions in early Earth would have been these there. And then they took them and they added electricity, so as a form of energy. And they were able to get out of this more complex organic molecules. So organic just means carbon-based. So the carbon in the methane was able to mix with the ammonia, the hydrogen, and the water, and you had more complex molecules made. These are all relatively simple organic molecules. So what this said was, hey, we had the conditions for life on this planet. That's what you want to remember. So the key to this, the take-home, is that we definitely had the conditions for life on this planet. So it's possible that life came from here. I mean, that's all this is saying is, hey, with this simple amount here, if you provide bond energy, you can make some complex molecules of which life is made out of. So that's what they did, and that's what this is about. So the only reason I told you that is because they sometimes, I don't know, they act like you know that. The beginning of the work starts here. So this is one. This is not a memorize. This is just a, okay. So somebody did an experiment where they proved that you can actually get something fairly complex out of something simple, and these were the conditions that were available. Making the point that, because all life as we know it is carbon-based. All life. Like, we just can't even wrap our heads around even some of the, I don't know, some of the weird little science fiction-y stuff. You know, they'll base life on different things, but carbon is what we base life on. So we have to explain, so we need to tell what the molecular diversity is and why it matters. That comes from the skeletal variation. So if you want to think about that, they're saying the way that the backbones or the way that the carbon parts can be put together. So the skeletal variation of carbon just means how you put your carbons together is going to provide the basis for the fact that we're carbon-based. Like, why are we carbon-based? Why aren't we something else based? So carbon, this is 2, and I have no idea how many pages this is going to take. I'm just telling you. So here's the deal with carbon. Carbon is the smallest, when you go on the periodic table, smallest size atom that has only 4 valence electrons. Small's powerful. Once you get big, you start losing the ability to have the atomic forces that are powerful enough to do what you want to do. Small, you have lots, because you're really tight and close to that nucleus, relatively. Now, we've got to remember, pretty far. But still, compared to the others, as they get bigger and bigger and bigger, they have less ability to form those really powerful atomic associations. So carbon, the reason why it's the king, so carbon, you're the king, how come, is this. We're going to draw a Lewis dot. I can share a pair here, and here, and here, and here. That's magic sauce right there. Everybody else can share less. So we want to write down, I cannot tell you how important that it is that you pay attention to what I'm writing. It is a test question. It is not okay to miss it. It is not. It's like not being able to describe what your best friend looks like, okay? It's just creepy. You should understand why carbon's important. So we're going to make a bold statement. Carbon, that's why I'm in all shouties, is unique. It's unique. Here's the reason why. I'm going to list them. The first is what I just drew right there. It has 4 valence electrons. The most possible. So we're going to write some things that means. First of all, that means that's the most possible number, most possible you can have. it means that it can make 4 unique bonds. Basically, it is the Swiss Army knife. It can do just about anything. So because it has 4 valence electrons and it's small, it's going to be able to make 4 unique bonds that are stable. It's small, so it has, I'm just going to put good, atomic, I'm going to put strength. I don't know how I want to word it, it's what I'm trying to tell you. So the atomic associations are powerful enough to form stable bonds. Why don't I just write that? Small, it has good atomic strength to form stable bonds. Because I'm pretty sure that life doesn't go on very good if things aren't stable, or if they readily become something that's in oxidized form, like not good. Okay, I'm looking at my notes because I want to be sure I get all these right. Okay, so first thing I would say, if I had to tell, hey, carbon's unique, defend that. Well, it's unique because 4 valence electrons. So in my brain, I'm picturing it's like a Swiss Army knife. You know what I'm talking about? The ones where you like, it's a can opener, it's a toothpick, it's a knife, it's like, what do you want carbon to do? I can do it. Carbon says I can do it. This is one of those days where, is this piece right here? Is this piece right there? Tacking. An eyeball. I get my hair done right before you come back. Every time I always think I'm going to G.I. Jane it, just whoosh, let it go. Drive me crazy. Okay, so here we go. Hey, carbon, you're unique. Yes, I am. Why are you unique? Nobody is the same size I am and can make 4 unique bonds because I have 4 valence electrons. What else can you do? Oh, I can bond with myself. So one of those bonds, or 2 of those bonds, or all of those bonds can be with myself. It's like a Swiss Army knife with the Swiss Army knife inside of it. Think about that. So you're just going to improve upon the fact that you can make many different bonds, and it can bond with itself. Okay, so I went and I got some molecular models. I'll go stop this in a minute and I'll bring them over. But how does it bond with itself? Like that. Isn't that the next question? Really cool. How do you do that? Oh, I can do that. Let's put a little note here because we're going to bring the cool thing about it. That's the third cool thing. It's how I do that. I can bond with a single bond, which I'll talk to you about all this. I can make a single bond with myself. Okay? So covalent, always covalent. Okay? In this case, not polar, because it's sharing with itself and it's an equal share. So I have a single bond I can make. I can, crazy enough, I can make a double bond. What? You can share more than one pair? I can make a triple bond. No way. Yeah, I can. That's insane. That's insane. And you don't think that that matters? Like you think, whatever, it's just 2 carbons together. Oh, ho, ho, ho. There's a ginormous difference when you have a single versus a double and triple bond. Totally different. Like, I'm going to tell you all about that. I'm going to put this little piece of hair on my face. Okay, here we go. So it can bond with itself. And then that leads to our third cool thing, which is how it bonds with itself. Then the fourth one, well, does it always bond with itself? No, it can bond with almost anything. So it's going to be able to bond with many different atoms. I always consider it to be an equal opportunity bonder. So it's able to bond with most anything. And when it does this bonding, the way that it puts itself together is lots of different like patterns, really, I guess, the structure. So I can bond with myself, which I'm pointing down here like you can see it. Down here. Oops, it's still not helping. Down here, bond with myself. I can bond with other things. So if you think of it more like Legos, you have the ability to put these together in a lot of different ways. And it has mostly to do with the carbon backbone. So, let's think about that. So the carbon, when they say backbone, they're just saying the carbon part, and then all of the other bonds that are out there left over, then they could be bonded with different things. But we're talking about the carbon backbone. It's going to be able to be in lots of different like structures. So carbon backbone, many ways. Now, I'm not going to worry about double bonds, single bonds. I'm just going to show you in general what I'm talking about. So one of those is straight. So you can have a straight backbone. You'd have your carbon. Okay, get where I'm going? That's a straight backbone. Or you can do branched. It looks a little bit like this, but it also has carbons coming off to the side. So you might have a carbon, branched. So not straight, but still fairly linear. Or you can have a ring. It's not going to be too difficult. I've told you I'm not worrying at all about any type of double, single, whatever, but you could have a ring shape. The number one thing to remember is, so this is super important. This is like a pivotal thing that we need to talk about. The difference between these, how many carbons did I do? Okay, so let's just put one more here. Okay, there's 6 carbons, there's 6 carbons, there's 6 carbons. These molecules do not behave in the same way because, here's the key. Ooh, purple's good. Change in structure, this is true for everything, equals change in function. So if you go from straight or branch to rolled up in a ring, the molecule does not behave the same anymore. So you have a change in the structure and you get a change in the function. Now every little piece of it might still be the same, but it's kind of like a Lego. I don't know how you were, but when Hunter got Legos, he would build whatever it was going to make, and then he'd build something else with it. And it did something totally different. Same pieces, but he built it differently, so it behaved differently. Same thing here. Okay, so we've now got a lot of reasons why carbon is unique. Small, has the ability with the small atomic size close to the nucleus, relatively close to the electrons, to form stable bonds, and it makes a bunch of them. How many? Most possible. 4. And it can bond with itself. It can bond with itself single, double, triple. We're going to talk about why that matters in a second. And it can bond with all kinds of things. Like it's not just stuck binding with itself, bonding with itself. And then it can make the backbone in a whole bunch of different shapes. So, right here, I am telling you flat out, this is a short free response question. You better be able to talk about it. It's like not being able to describe your best friend. It's creepy. Like you need to know why carbon's a big deal. Carbon's the king. Why? Let me tell you. Okay. All of this now, as we're starting to see how that the carbons put together, and they go together in different ways, you emerge, just like we talked about before, with properties you didn't have before. Like individually, carbon, still cool, but when you start putting them together, okay, emergent properties. So we're going to talk about isomers. So I'm just going to do like that because we're here at 3. Because this, to me, deserves to be its own blocked off thing. It is what the chapter is about. I don't know how to stress this anymore. What's an isomer? You cannot. Describe the specific isomers below. So there's names of isomers based on what an isomer is. We don't even know what that is yet, so let's go figure out what that is. So I'm going to come over here. This is part 3, and we need to know what an isomer is. So it's the same parts rearranged differently. So let's see how we can say that. So isomers, they're going to be molecules with the same atoms and the same number, the same parts and pieces, but they're rearranged differently. What did I say about that? What did I say about change in the structure? Change in the function. So yeah, your parts are the same, but you put them together different, so they're going to do something different. So molecules with the same atoms, but different structure. So we're going to put in here. So they're put together differently. Okay? That's what an isomer is. So when you start talking about glucose, there are isomer forms of glucose. So are they different? Well, they're all C6H12O6, if that's what you're asking. But when they're put together differently, there's a reason why that is. So most of the time it's branched. It can be in what's called a chair. But when it goes into water, it becomes a ring. So it's going to always lean toward the most stable form based on the conditions it's in. So isomers are all the same atoms, C6H12O6 for glucose. But can you put all those pieces together differently? Yes, I can. Is it still glucose? Yes, it is. So we would have to preface with the reasons of, but there's a reason why that they're in different forms. Aqueous, water, ring. Not aqueous, tends to be branched. Okay? So let's talk about what the names of these are like, because they gave us some specific ones. So the first one is the difference in the way you're put together is structural. So that's what I just described. So let's come over here, structural isomer. This is going to be exploiting what carbon can do with, hey, I can be straight, I can be branched, I can be a ring. So it's the same parts, the same atoms, but they're going to have a different backbone with carbon. So the same atoms, but they can be straight, branched, or a ring. So you see we're speaking here with these isomers about carbon. Carbon's part of this, it has a backbone, and if its backbone is straight or branched or a ring, those are structural isomers of each other. So when it can move between forms, that's the other thing that's super cool. Let's put that here because that's super cool. It can move between forms. So just like I said, you might have glucose that's branched and we'll talk about what you taught there, be D-glucose, but whatever. Glucose is branched. And then you move it into water. So move to water. So you drop glucose in water, and it rolls up a ring. So you would let someone know that this is glucose, and they put an AQ here in an aqueous form, and it rolls up because water's polar, it's going to roll up into a ring where it's in its most stable configuration. Or we could get that word out. Cool with this one? This has to do with all of the different backbone shapes. Structural. This is one. We've got geometric. I'm going to go grab some of those molecular models. Alright, so I went and got my friends. So, little just models with carbon. And at this point, let's focus on this little guy. Notice that you've got a double bond here, and you've got a single bond here. So I'm going to pull this off, and I'm going to sometimes I don't want to work chicken. Okay, so I'm going to have a carbon-carbon bond there. When you have a double bond, when you have a double bond versus a single bond, notice that this is a single bond. Do you see how I've got free rotation around that bond? So everything I've got there is, okay. If these, let's say, were different colors, and I said, ooh, look, white's on top. Oh, I'm going to pretend. Red's on top. Whoa, nope, blue's on top. Oh, white's on top. This is going to have free rotation. There is no spatial way to say, hey, look, we're on the same side. Nope, we're not. There's no way, because it's free movement around that single bond. Double bonds, nope, lock it in place. So now you do have the ability to speak to position. So let's look here. So you've got two double bonds here. So this would pretty much be carbon dioxide if you wanted to let that be. And these double bonds lock whatever that's attached to the carbon spatially down. So you're able to have the fact that you can see this is linear. And it's not like that this can move or go anywhere. It's locked in space. So double bonds, you have no rotation. And you're able to talk spatially about where things are located. Single bonds, you may not. You cannot do that because there's rotation around this single bond. So whatever's out here attached, not only is this freely rotating, but this is freely rotating as well, because these two carbons are single bonded. So there's all this free rotation around that bond. Triple bonds look like that. They definitely have the ability to lock pieces of the molecule, atoms in the molecule, spatially down. Okay, so I'll tell you all that for the next, which is geometric. So, I messed up my whole thing there. Alright, so geometric isomer. Isomer means same parts put together differently. This time we're not talking about the backbone, we're talking about the location of these in space. So this is going to be where that you have the ability to have a reference on each side. So this is what I'm saying. So if I have carbon double bonded to carbon, which I don't have, but if this would be a double bond. This is rigid. It cannot rotate. Whatever I put up here at the top is going to stay at the top. It's not able to spin, so this is a rigid bond. So now I've got a cool thing. I can talk about whatever's attached, because I've got 1, 2, right? 3, 4. And then on this carbon, I have 1, 2, 3, 4 different bonds. There's definitely we're on the same side or we're across from each other. So if you have, let's just say your hydrogens are both on the same side, and then you've got what's called R groups. R is just a shortcut way to say whatever is attached here. So some group is attached here. We call it an R group because we don't know what it is. Some group, variable group, is what this is technically called. If these are on the same side, this is called cis. Cis means same. I can make a geometric isomer with 2 H's, 2 carbons, and 2 variable groups, and I can put it together differently in space. So I'm going to have the same double bond with the carbons. I'm going to have the same 1, 2, 3, 4 for this carbon, 1, 2, 3, 4 for that carbon. That part doesn't change. But now I'm going to put these across from each other. So the R group is across the double bond from the other one, and the hydrogen is across the double bond from the other hydrogen. This is called trans, which means across. So geometric isomers are about placement in space of the groups that are attached to carbons that are double bonded to each other. And so this trans version isomer of the molecule does not behave the same as the cis. Okay? Especially if that R group was big, you'd have 2 big groups over here, and hydrogen is the tiniest thing you can have. So it'd be super heavy on this side, or if you had small and it could be huge and could be small, you'd be balanced more here. So they're going to have a different behavior because they're put together differently. The number one thing to remember: change in structure, change in function. You cool with that one? Okay. So enantiomers, these are, I always think of gloves. They're like your hands. So your hands are mirror images of each other. Do you have 4 fingers and a thumb? Yes. On both hands? Yes. So if I give you a glove, can you wear it on either hand? No. Why? Because the position of my thumb relative to my fingers is different. Oh. Really? Yes. So these are mirror images of each other. And this has to do with something that we should go back and add for carbon. So the reason why this occurs, so reason why, and I'm going to put pink gloves. The reason why they're different is because carbon forms a tetrahedron. So a tetrahedron, that's why these things are really aggravating, is that. That's a tetrahedron. So if I put something here, it is in a different place relative to the others than the mirror image of it over here. So the way to put this is it's positionally, yes, positionally, I made that word up, distinct forms. And what it really is relative position. So this is of all of what's attached to the carbon. So if I think about this, I'm going to remember that my relative position of my thumb to the, I got to put 5, that's not going to work, to 4 fingers is what we're talking about. Because that's what you get. Your thumb relative to your 4 fingers is different. And even when I do this, there's a sidedness that comes in. So yes, now my thumb's in the same relative position. Look at that from where I've been writing. Same relative position to my fingers, but it's the back side of my hand. So this tetrahedron gives carbon the ability to have this positional placement that's different and that varies. And what we usually do with these, let me highlight these real quick while I'm thinking about it, is we think about this a lot of times for drugs. So, you know, we have Eastman here, but if they put some, if they're trying to make a molecule, let's go that route, they will make both forms. They're usually called here, they're usually called the L form or the D form. And they'll make both. And for us, only one is biologically active. And the reason is this. Because they're mirror images of each other, the molecules have to fit into an enzyme pocket, into the active site. And just like what my right hand would fit into, my left hand will not. One of these forms will fit and the other one will not. So they're called forms. One is bioactive, and the reason would be because it fits into the enzyme, which those are like the little machines that do work, pocket, the part like the glove, so the enzyme active site. The other one won't. The other one won't fit. So drug companies all the time make this. You have to be really careful, because a lot of times what you'll do is you'll buy vitamins and you think, oh, or some type of supplement, and you're like, that's pretty cheap. I'm going to get that bottle. Well, only half of it's bioactive because they put both forms in there. If they take one form out, it's more expensive. Like if you get L, like tyrosine, then it's going to be the form that you can use. Or if you just go by tyrosine, it's going to have both. So enzymes are only going to be able to work on one or the other, and that's going to be the one that's bioactive. But if you just manufacture them, you make them both, because the probability is the same. So it'd be like if I made hands, the probability is exactly the same that my thumb went here or my thumb went here. But my hands are different. My hands are different. And so I couldn't put them in the same glove like the enzyme. We're good with that part? Okay, does that finish that strip? It does. Okay. We're going to add some little videos. Don't hate me. I'm trying to make this to where that whether you're here or not here, or we're virtual for part of the, I don't know what we're doing. I'm going to hang with Canvas, because I'm going to take care of you. That's my job, because you're the best.
No matter how green, how furry, how small, how big, or how weird, all known life forms are based on the chemistry of one element, carbon. But why carbon? And if we look out to the stars, could there be a fundamentally different kind of life out there? Questions like these are really hard to answer because we only know life on one planet, ours. Life here on Earth is fundamentally chemical. DNA and proteins are both super long, basically infinitely variable polymers based on carbon. Fats and sugars are based on carbon. Most molecules cells use to signal each other are based on carbon. We don't know of a single life form that doesn't use carbon as the basic scaffolding of its own existence. Of course, life needs more than just carbon. A lump of pure carbon, whether graphite or coal or diamond, is not alive, but carbon does seem to occupy a special place among the elements, at least here on Earth, and probably on other Earth-like planets as well. Let's take a look at some of the reasons why. First of all, carbon is fairly promiscuous, meaning it can form bonds with a wide variety of elements. In this drug, carbon is married to other carbons, oxygen, chlorine, nitrogen, sulfur, and hydrogen. Secondly, carbon can bond a bunch of different ways. It can form incredibly stable bonds with some elements like hydrogen, and it can form less stable, more reactive bonds with others like oxygen. Carbon can form tetrahedral, aka diamond-like shapes, it can form flat rings, and it can even form molecules that look like those rickety beach lounge chairs you constantly fell off in the 90s. And with help from other elements like hydrogen, nitrogen, oxygen, phosphorus, and sulfur, carbon can form the backbone and side chains for the incredibly long polymers that are key to life here on Earth. But it's not inconceivable that, somewhere in the universe, elements other than carbon could be the building blocks for life. Silicon, right below carbon on the periodic table, and therefore with some similar chemical properties, has popped up as a potential alternative. Silicon shares a lot of similarities with carbon, including its ability to bond to oxygen and to create long polymers. But it has some major setbacks. First, silicon struggles to create multiple bonds because of its size, and the bonds it does make are much less stable. But what really puts the nail in the coffin is that when oxidized, silicon creates a solid, silicon dioxide, otherwise known as sand. Whereas when carbon is oxidized, it creates a gas, which is easy for plants to access and make sugar from. While silicon has stoked the imagination of science fiction writers, the scientific community has yet to find enough evidence to suggest the potential of silicon-based life. Some life can already use silicon though, like in these
Hey, it's Professor Dave. Let's talk about carbon.
He knows a lot about the science stuff. Professor Dave explains.
Hey, this is Mr. W from Science Music Videos, and this video focuses on carbon, the central element in living things. As you learn biology, you'll learn about molecules like phospholipids that make up membranes, like DNA that stores genetic information, and like hemoglobin, which transports oxygen. All of these molecules are based on carbon. Here's why. Carbon is element number 6 in the periodic table. It's number 6 because it has 6 protons. It also has 6 electrons, and in the most common isotope of carbon, 6 neutrons. Think back to the octet rule. The first 2 electrons go into the first orbital. That leaves 4 outer or valence electrons. Those valence electrons readily form covalent bonds. If you need a review about covalent bonding, go to sciencemusicvideos.com. The link is right below. Here's carbon forming 4 covalent bonds with 4 hydrogen atoms to form methane. If you can form 4 covalent bonds, you can form chains. You can form rings and also branched molecules. Here's another thing about carbon. With 4 valence electrons, it easily forms double bonds and even triple bonds. This is a chain of amino acids, which are the building blocks of proteins. Notice the repeated double bond between the carbon and the oxygen. It's the basis for one of the chemical interactions that causes this protein, hemoglobin, to fold up into its specific shape, and that makes its function, which is carrying oxygen, possible. It all comes down to carbon and its 4 valence electrons. What's the takeaway? By weight, we're mostly water, but carbon is what makes possible the molecular complexity that life is all about.
By the end of this video, I want you to be able to tell the difference between a chiral object and an achiral object. So I'm going to show you an object, like this matchstick, and its reflection. And the question you have to ask yourself is the object identical to its reflection? In this case, the answer is obviously yes, so I put an equal sign. And our next object is a lighter, and there's its reflection. And by rotating the reflection, we can very easily see that again the object is identical to its reflection. Now moving on to some kind of hypothetical molecule. We have the object and its reflection. And again, we can very easily see that the object is identical to its reflection. Now changing one of the balls to blue, we ask again, is the object identical to its reflection? And the answer is yes. So changing another of the balls to blue, we ask again, is the object identical to its reflection? And again the answer is yes. Changing another of the balls to yellow, with a little bit more rotation, we see that the object is identical to its reflection. So changing the final ball to purple, we have the object and its reflection. And what I'm trying to show you here is that there's no way that I can rotate the reflection of the object so that all the colours are in the same place. In other words, the object is not identical to its reflection. And this is what chirality is all about, when the object is not identical to its reflection. So we have another object and its reflection. And there's no way that I can rotate the reflection so that those two objects are the same. So they are chiral objects. Again, we have some made-up molecule. And this made-up molecule is achiral. And finally, we have the famous example of hands. And my left hand is not the same as my right hand, which makes my hands chiral. So to sum up, a chiral object is not identical to its reflection, and an achiral object is identical to its reflection. Okay, so I hope you now understand the difference between chiral and achiral objects. If you liked it, don't forget to subscribe, because I'm going to be making lots of biochem and neuro videos over the next few months.